Okay, so hello kids. Any of you who know me from Biochem Biology 2200, I'm wearing my iconic red shirt and khaki pants. We're going to talk about what we do in the lab. If you're taking 2200 now, a lot of your TAs are from my lab, so you'll know them. We work on animals that are alive, and then they're dead, and then they come back to life. So life in limbo. It's also called metabolic arrest, where instead of running your metabolism all the time, using up fuels, generating heat, producing end products, you just turn off all 50,000 or so reactions in the cell at once. It's the at once part that's hard. You can't do this. Many groups around science are interested in this. We get funding from the European Space Agency, who are interested in turning off humans and sending them into interstellar space. For organ transplants, they're interested in organ transplants, as well as a little bit of money about concussive injuries, where you have an injury and then you try to turn off the person injured and then get them to safety. So there's a website for us, and this talk and others are on the website. So let's see the next slide. So the kind of animals we work on, the normal ones are animals who can turn off their metabolism, but to tell you the truth, humans are the only ones that can't. So I'll show you animals that are different kinds of mammals and other lower animals, and today we'll focus on the mammals. So you might know about hibernation. These are 3 animals that hibernate in the cold. So when it comes winter on the Carleton campus, the groundhogs disappear, the chipmunks disappear, the ground squirrels disappear. And that's hibernation. They've dropped their metabolic rate, they're living in a suspended state. But other animals can as well. Some animals can live without any oxygen at all, so 0 blood oxygen, and the highest form of life that does that is a turtle. Some animals estivate, so they get really hot in the desert and then they dry down and become crispy critters. So underneath the desert floor are toads and frogs and snails that have turned off their metabolism and dried down strongly. And then there's the animals that can freeze. So we study frogs and turtles that can freeze solidly, and insects that live in Canadian climates where it gets to be -20 and -30. So all sorts of animals do it. You're the only one that doesn't. We'll focus in on the mammals. Next slide. And here are the mammals. You're somewhere on this chart, hopefully in the primate section. So there was a stem mammal, and that stem mammal could obviously hibernate. Mammals came in evolutionary time from therapsid or anapsid reptiles. So we were reptile and then we became a mammal slowly. Probably the most ancient animal on the planet that still hibernates is called a tenrec. We study tenrecs in Africa, and they're a specialized form of life. They hibernate in a very ancient way, and we're studying them in order to see how hibernation developed. But in every group of mammals, there's some animals that have remembered hibernation, and most animals in that group haven't remembered. The most commonly studied are the rodents, the rodentia. We have papers on marmots and ground squirrels and dormice and things. And the primate that hibernates is a lemur. So we'll show you some lemur data. And then the warm-blooded animals that get cold in Canada include bats and other little creatures. So the next slide is the animals that you've seen that exist around here that we study regularly. These are hibernators in the cold. So you see them in the summer, then they go underneath the ground or in a cave. The bats are in caves everywhere. The populations here have fallen quite strongly because of white nose. They have a fungal system. So these animals are going to be warm and active, then they're going to cool down. So let's see that, the next slide. These are animals that are marsupials, live in the cold and cool down. These are monitos. They live in the mountains of Chile. When you look at mammal evolution, marsupials came before the placental mammals. And so we were interested in whether or not marsupials knew how to hibernate, and if they hibernated in the same way as the placental mammals, like the mice and the ground squirrels and the bats. So next. This is how animals that hibernate in the cold hibernate. They disappear for a long period of time. You don't see them, they're hidden. But even when they're hidden, they don't keep their body temperature really low all the time. They cycle between a low body temperature and a high body temperature, and a low body temperature and a high body temperature. So this is a thermal probe in a ground squirrel. And so what happens is they start at 37, and then they begin to have shorter periods of hibernation, then longer periods of hibernation. So during the winter, even though you don't see it, underneath the ground for 2 weeks, they're cold. They're lying unmoving, just like icicles. Then they warm up for a day or a day and a half at a time. And at the top of their warm cycle, they're aroused. But they don't run around, they don't eat, they don't drink water, they sleep. So when the animals are warm in the middle of the winter, they sleep. Their brain goes into REM sleep. And at low temperatures, their brain is really jangled. So we've done a bunch of brain studies on how the brain works and what kind of molecules they build up. So we can sample animals that are warm, that are going into hibernation and getting really cold, that are coming out of hibernation, and then that are warm again and sleeping in their den in the winter. Animals come out of hibernation by warming with brown fat. So this body temperature rise is all driven by brown fat. You can see our papers on brown fat. Brown fat's turned on. It only generates heat because it's uncoupled, and boom, you're warm again. But you're not running around. You're not a summer animal. You're still a winter animal who's just getting your brain organized. So next slide. This is a cooling curve of hibernators that we did a long time ago, and it shows that animals turn off their metabolism first, then they cool down. This is going to be true of any hibernating animal, even if they hibernate in the warm. First, you turn off your metabolism, and therefore you're not generating any energy, and then you cool down to ambient temperature. So the red line is the temperature of the animal. Starts high, high metabolism, everything's fine, then boom, the metabolism drops off, the blue line, and then the temperature drops off and comes to whatever the air temperature is. So if you're a tiny, tiny, tiny little animal like a bat, your body temperature falls almost instantly, Q10s of 15. The bigger you are, the longer it takes you to cool down. But eventually, all these rodents and all these bats cool down. A marmota is the marmots that live on campus. So if you've seen groundhogs, these are groundhogs. They turn off their metabolism, then they cool down to the temperature underneath the ground on the Carleton campus, which is roughly 5 degrees. If you are big enough, you don't have to cool down. It takes you so long to cool down that you don't really cool down. I'll show you an animal that does that. It's just too big to cool. And if you hibernate in the warm, your body temperature only falls to air temperature. So if you're hibernating in Madagascar, or if you're hibernating in the depths of the jungle, then you're fine. You only come to the temperature of the jungle. Okay, next. Boy, I wish I could use that button. There are publications on how hibernation works. So these are review articles. These aren't regular papers with a whole bunch of data. These are review articles, how you link hibernation. So hibernation is linked to longevity. Animals that hibernate, like bats, live for 30 years. Bats that migrate live for 3 years. They Hello? If you can hear me now, you're okay. I was muted by somebody. I'm really sorry about that. There's little I can do.
We can hear you now. It's all good.
Oh, good. So these are review articles. You can see them on PubMed. You can go to our website and see them. They're how hibernation works, how it links into longevity, how it links into stopping of metabolism and stopping of disease, how you can use hibernation as a tool in order to turn off metabolism for long periods of time. Okay, next. Oh, these are a warm hibernator that go into torpor at really, really high temperatures. These guys live in Madagascar. They're a type of primate. So in Madagascar, it's not so much warm and cold, it's dry and wet. When it's wet, they got a lot of food. When it's dry, they have no food, and so they go into a metabolic depressed state. They hibernate as groups. They actually hibernate in big trees, in holes in big trees. And this is the lemur. It's a smaller lemur than you're used to. So next, it's bears. Now, you know bears as being big, and you know bears as living in the snow. So what do bears do? Bears are so big that they don't cool down. So if you go and find a hibernating bear and check its temperature, it's still 30 degrees. It's not 37, it's 30, but it's not down to 5. So they hibernate in the cold. Their air temperature around them is snow temperature, 5 degrees. They're so big and so much fat and so much fur that they can keep their body temperature warm. They hibernate like a lemur because their internal body is still warm. So bears, lemurs, ground squirrels, there's different ways of hibernating in different temperatures, but their metabolism is going to be the same. When we go over all the chapters that are in the biochem textbook, you're going to find all animals have a common way of hibernating at the molecular level. Okay, next. So here's the deal in words. There's a metabolic rate reduction of every animal. Every animal drops its metabolism really strongly. It stops doing all the things, all the organs, even the brain turns off. And then what happens is their body temperature goes toward the temperature they live in. If they live in warm temperature, you're fine. If they live in cold temperature, you're fine. And you turn off processes in hibernators that you can't turn off. So even though lemurs are a primate and you're a primate, you can't do what lemurs do and you can't do what ground squirrels do. If I turned off most of your genes, if I turned off your cellular processes, you would actually die because there's no way you can turn them off in order. There's no way you can turn them off in an organized fashion. And that allows our students to study almost any aspect of cell function and any organ in the body, including the brain and the heart, to see how you turn off a brain and a heart. What we're going to focus on today is one of the early mechanisms, maybe even the cause of hibernation, called microRNA. MicroRNA is part of epigenetics. It's a relatively new field of study, and we find that this probably starts hibernation, and then a whole bunch of things happen downstream with hibernation. So we'll sort of look at epigenetics as a central controller, the control that turns off everything. All metabolic rate depressed. Next. Oh, now the first thing that happens is this microRNA thing. And one of the first things we see is control of the cell by protein kinases. So you have between 750 and 1,000 protein kinases. There are two big families. One are called stress-activated protein kinase, and ones are called side messenger kinases. If you took 2,200, we sort of introduced them both. Even though you have almost 1,000 of them, and over 3% of your genome encodes for kinases, in hibernation, most turn off. Most, boo, just like everything turns off, and about a dozen turn on. These dozen that turn on actually reach out into the cell and turn everything else off. So it's like somebody waking up and turning off all the lights. And the protein kinases that are turned on are the next line, the third line. They're not important here. I'll sort of introduce them a little bit, but imagine they're kinases. They're going to phosphorylate other enzymes. They turn on and turn everything else off. These kinases are present in the hot bears and in the cold ground squirrels and in the bats. These same kinases deal with lemurs and monitos. And they're also involved in all those other weird animals, the anoxic turtle, the freeze-tolerant frog, the estivating toad in the desert. They all use these kinases turned on in order to turn everything else off in the cell. So the next slide is a picture of a cell. Yay! This is what your cell does. So right now, you're 37 degrees and you're generating heat. You're just running all of your processes. You're using up fuels, you're making end products. You're turning on and off your genes in the nucleus. You're making messenger RNAs, which are really expensive. You're then making proteins from polysomes. That's way expensive. You're pumping ions, out with the bad ions, in with the good ions. You're pumping and regulating calcium. You're running carbohydrate, amino acid, lipid pathways, and each one of these pathways generates a little bit of heat, generates some ATP, uses some ATP, and of course, most of your energy, most of your heat is made and used in the mitochondria. So if you look at this picture, what you have to do is turn everything off at the same time. And short of shooting you full of heroin, I really can't do that. And heroin at those levels isn't even safe. So what the hibernators have done is taken 700 to 1,000 different enzymes, identify 12, turn those on. So the SAP kinases turn on, and they reach out and turn off the nucleus, off the pathways, off protein synthesis. The second messenger kinases turn on, cyclic AMP, cyclic GMP, calcium, IP3, and they turn off pathways, especially the central metabolic pathway, and they reach into the mitochondria. So a lot of our mitochondria is turned off by these second messenger kinases. It's a great system because you only have to turn on a couple of things. The total amount of energy involved in turning on these kinases is a fraction of 1%. So you were 100% active, generating heat. Now you're less than 1% active. And this is how you can turn off your metabolism. We can modify some of these pathways by using drugs and chemicals. So there's molecules made by drug companies that turn on and off these pathways. And so we do experiments where we isolate organs and look at that. So the next slide. Ooh. A few kinases are activated, turning off thousands of different processes. What you've also got to do, point 3, is you've got to turn off genes. You can't be making messenger RNA because it's too expensive, but you turn on a couple of genes. So 1% of your genes are turned on, 99% are turned off. And that can't be done with kinases. So kinases work in metabolic pathways. They also work with some transcription factors. But if you want to turn off all your genes, you have to go to the wall. And that wall is epigenetics and microRNA. So the trick here is that the cell is controlled. The brain of the cell is in the nucleus. And that nucleus is going to be controlled by epigenetics and microRNA. So the next slide. Oh, yay, it's epigenetics. You know this, you love it. It's now covered in molecular biology courses. It's covered in first year in some places. We cover it in Biochem 2200. If you're a hibernator, control versus hibernated, you have the same DNA. You're not allowed to change your DNA. Everything has to be the same, but you can alter that DNA, not the sequence, but the activity. So you can turn off the activity of your DNA in various ways. You can turn off transcription, you can turn off translation by reorganizing around this epigenetic core. Epigenetics used to be like unknown and impossible, and now it's being involved in everything. You'll see new healthcare things to do with diseases and epigenetics, to do with the regulation of various kinds of problems in the human body. The mechanisms which we'll cover is DNA methylation. You methylate DNA, and that changes its activity. You modify histones. Histones are proteins in the nucleus, and there's lots of them. The principle is you covalently modify histones, and they grab up DNA and squash it all up. And so you can't get any transcription. So histone modification either opens up and makes more transcription or closes down and makes less transcription. And the third great mechanism is non-coding RNAs. These are called microRNAs. They're non-coding. They're tiny. They're never going to be messages. They're never going to be transfer RNA or ribosomal RNA. They're specifically made to be regulators. That's all they do. They regulate something, then they're broken down by the cell, then you make more and regulate something. So next slide. Oh no, well there we are. Is the conceit in the middle, the idea in the middle, and then the ways it's regulated on the outside. So ATP expensive. You're making and breaking ATP, you're making heat. Hibernators stop that. No matter what their body temperature, they've turned off their metabolism. And the way you turn off your metabolism is you methylate the gene, you modify the histones, or you regulate microRNA. There's a fourth group of things that happen. It's in the lower left-hand side of this slide. We're not going to cover them today because they're complicated, but if you look at our papers, we've got a bunch of papers on them, forming stress granules, regulating metabolism, and RNA binding proteins. So we'll look at those three. Next slide. Oh, next slide. Does DNA methylation happen? Yes, it happens everywhere. It happens in the hibernating mammals that we're looking at. It happens in the frozen frogs and the turtles that go anoxic. It happens everywhere and all organs all the time. It's difficult to study because it's so expensive. Each one of these papers is outrageously expensive. You need big grants. And what we do now is we simply sequence an entire genome. Then we take the hibernating version and sequence the entire methyl genome. So you just do mindless DNA sequencing. There's miles and miles of DNA, but they're very powerful computers. So we have supercomputers in the lab, and we let those supercomputers say, where have you methylated? What have you methylated? Come on, come on, come on, tell me. And we do gene and DNA sequencing by this new GridION method, which is a nanopore method. So what we can tell you is that if you look at the papers, DNA is strongly methylated and shut down, same sequence, just shut down. So the next slide, this slide may move, I'm so excited. Next slide. It doesn't. So DNA methylation you see everywhere. You see those papers which cover all of our animals. And the second one is histone modification. You want to take the proteins that are in the nucleus, covalently modify them, methylate them, acetylate them, phosphorylate them, change their way of grabbing up the DNA and making certain the DNA shuts off when these proteins are covalently modified. So next slide. Are histones modified? Yes. Histones are modified everywhere, in every animal, in every tissue. So everywhere we look, you can look at these histone modifications. What happens is histones are basic proteins, lysine, arginine, and those are modified by mono, di, and trimethylation. You have to look at the enzymes that modify the histones. Then you have to look at the marks on the histone. They're called histone marks. You look at the marks by antibodies, and you look at the enzymes by measuring their activity. So we have kits, and Liam Hawkins has developed a new way of measuring histone modifiers. There's three big classes of enzymes that work in the nucleus, and ones are readers, writers, and erasers. So writers are the ones that put the histone marks down. Then the readers go along the DNA and say, should I turn the gene on? Should I turn the gene off? And then erasers just get rid of all the marks again. So if you imagine a ground squirrel, when it's hibernating, when it's cold, its histones are marked. It's methylated and acetylated and phosphorylated. Just then, as soon as, remember how they warmed up in order to get their brains straight? Boom, the erasers just come and get rid of all the marks. And then a day later, boom, the writers come back and turn it off. So this is histone methylation during hibernation and then the release of hibernation. And it's not only mammals. This happens in giant squid that live at the bottom of the ocean. This happens with insects that are warm during the day and freezing cold at night. It's a mechanism by which an animal can turn everything off. Ironically, you have histones, you have erasers and readers, and you have everything in place that could do this, but you can't organize it correctly. So when we take a human cell and cool it down, it just acts like an idiot. It doesn't hibernate. All these enzymes are just running around doing stupid things. And if we take one of your organs and try to turn it off, so you take a heart for transplant. We have a heart and stroke grant at UCLA with the Gottlieb lab. And if you just take a human heart and try to cool it down for transplant, it just goes nuts because it doesn't have the instructions on which to do it correctly. So the next slide. Oh, the next slide. We talked about the two green things. Genes are methylated in their promoter regions, also in intron exon overlaps, also in CPG islands. The details are unimportant. What happens is when you methylate a gene, you can't get messenger RNA, so you shut down. Then histones, global changes in all the histones of all the cells of all the organs, you just shut the thing down. So it used to be expensive to run a hibernating primate. Now what you can do is turn off its nucleus and you save tons of money. You save 60 to 80% of your ATP use. Boom, your metabolic rate is going to start to drop and drop and drop. So the next slide shows the next thing. The next slide talks about microRNAs. What we're doing now to orient you overall is you're using epigenetic programming to remodel chromatin. Now, what happens is you use your metabolic pathways, you use molecules you've eaten, you use cues from your environment to make a bunch of metabolites. These metabolites then act on the readers, writers, and erasers in order to change the DNA. So it is a very complex area, but you have an epigenome. So you have a genome, all your DNA just read out by these big machines. Then you have an epigenome. Where are the marks? What are the marks? Who's being modified? So this is the overall principle here. Your cells and the environment are reaching into the nucleus, altering your DNA, and then, of course, your DNA stops its function, slows its roll, and doesn't use as much energy. So next slide. The next and the last one that we can cover here is microRNA. So microRNAs are going to be small RNAs, and what they're going to do is help turn off the nucleus. They are going to reach into the cell after they increase during hibernation and turn everything off. So we have so many papers that you're just going to have to look at PubMed and look at a couple of the reviews. They work everywhere. So the freeze-tolerant frogs, sea cucumbers, they work in sea cucumbers when they get too hot. They work in lemurs. They work in frogs, toads, turtles, ground squirrels, and that little thing is a little tiny crab that lives at the bottom of the ocean. It works in weird things in the ocean. So the next slide talks about microRNA. Turning it all off. So there's about 1,000 microRNAs. We've discovered species-specific microRNAs that were never known. It's fairly easy so long as you have the equipment, and then you look at these microRNAs. In general, they increase when metabolism decreases. They increase and then they reach out to turn things off. So you can look at the review articles. It's a general principle. They differ in type and amount in each organ. So the microRNAs that turn off the brain are slightly different in sequence than the microRNAs that turn off the heart. But wow, once you know the microRNAs, once you measure them all, then you're okay. So what we do is we use another type of omics, right? We did DNA omics where you just sequence the genome, but we also do microRNA omics where we measure all the microRNAs and then organize them in patterns. So the next slide goes into how you study and do microRNAs. MicroRNAs are small. They're relatively easy to sequence. You just have to have a large number of sequences done. So this is expensive because machines do it. They're really highly conserved. Almost all mammalian microRNAs, all 1,000 of them are the same sequence. You have the same microRNAs as the lemur. You have the same microRNAs as a cow. You have the same microRNAs as a mouse. There's a lot of similarity. And they seem to reach out and regulate individual genes and turn them off. So to be conservative, 85% of genes can be controlled by microRNA. And by controlled, we mean shut down. I do not know of a current human disease that's studied that isn't microRNA controlled. Alzheimer's, all sorts of diseases of the brain, and as well as heart attacks, strokes, diabetes. It's crazy. There's a microRNA involvement everywhere. It makes it interesting in that microRNAs weren't known. Now microRNAs control everything. So you find a situation with microRNAs where if you look at the number of papers a year, it just goes up exponentially. It's just crazy. Everybody's doing it. MicroRNAs are of many different sequences, but their general role is to block translation of message RNA. That's just what a hibernator needs. Why are you making message RNA into protein? It costs you energy. Nobody cares. Stop that. And it also targets messages for degradation. MicroRNA lies along message RNA with its associated proteins and then just cuts them up so you don't worry about it. So the role of microRNA is to turn things off, exactly what a hibernator needs, and a hibernator can turn everything off and you can't. So the next slide shows how it's done. So this is a metabolic pathway. This is, you can actually isolate the enzymes and study them that use this pathway. It's just a weird pathway. It's not like glycolysis. So in your genome are the sequences for all microRNAs. So somewhere in your genome, there's 1,000 different sequences for 1,000 different microRNAs, from microRNA 1 to microRNA 997b. What you do then when you want to make microRNAs is you copy those up. So what you have is a transcription factor that makes the microRNA. Then you polish the microRNA, send the microRNA out. You then dice and slice the microRNA into a mature microRNA. Then that microRNA grabs up some proteins called a RISC complex, and then it does its job. So you're starting from the right-hand side of this slide, where in your genome are microRNA sequences. When we sequence an entire genome of an animal, and we've done ground squirrels, we've done turtles, we've done sea anemones, we've done squid, what you could do then is you could have machines go up and down the DNA and say, hey, will this make a microRNA? So you can find brand new, never before discovered microRNAs and then study them. It's sort of cool, and it's all done with big machines and money. You feed money into these big machines like you put a dollar bill into a Coke machine, and then instead of Coke, you get sequences. So there's enzymes, DROSHA, dicer, exportin, and then finally, on the left-hand side, you get a mature microRNA. It's ready to go. It grabs up its proteins and shuts things down. So 2005 is when this was discovered. So this seems like a long time ago for you people who were born in the year 2000. But scientifically, this has grown crazily over time. This was a guy guessing in 2005, and now everybody's doing it. It's like a dance craze. Okay, next slide. This is how you do it. When you take a ground squirrel, what you find is its muscles, even though it doesn't move for 6 months at a time, don't atrophy. If I took you and put you in a hospital and put your legs in a cast, in a couple of weeks, your legs would look spindly and awful. You'd lose all the muscle. If I put you in a situation where you couldn't exercise, your heart would get weak and horrible. But what a ground squirrel does is turn on hearts, make them big and strong. They keep muscles from atrophying by making microRNAs that keep the protein levels up and strong. When a frog freezes, it builds up 400 millimolar glucose, which is like 100 times what would kill a diabetic, but it also has microRNAs that regulate its pancreas, just like microRNAs regulate the pancreas of ground squirrels. So you measure all the microRNAs, then you look at their functions, and then you put together this big ball of 400 change, and overall you can tell every metabolic pathway. You can tell every process in a cell by just looking at the microRNAs that change. That's how powerful they are. If you have 5 microRNAs that turn off a pathway, that pathway is done. Just, it's not going to come back until you warm up. So microRNAs are that powerful that you can measure them and know what the cell is going to do. So the next slide is a slide of the microRNAs, not of a placental mammal, not of a bat, not of a squirrel, not of a bear, but of an animal that lives in South America that isn't even a placental mammal. This is a marsupial. This is related to kangaroos and stuff. And here's the thing, exactly the same microRNAs. So this torpor-sensitive microRNA phenomena is something that was built up in all animals above reptiles to turn them off eventually in bad environments. We are now studying the tenrec, which is the stem mammal, and we wonder, is this the same? Nothing is known about tenrecs. You don't know about the microRNAs, and you don't know about their pathways. And the way in is you just measure all their microRNAs. Are they exactly the same as a squirrel? Are they exactly the same as a bear? So this shows you the power of microRNAs. They do the same thing everywhere. They activate a pathway called mTOR, and mTOR is an insulin pathway that regulates the use of fuels. They activate MAP kinases, which are one of the kinds of kinases, only a dozen that are turned on. So you say, how come there's 750 kinases turned off and 12 turned on? And the correct answer is microRNA decides. And there's also tissue-specific responses that are the same in a mammal, and same in a rodent, and same in a bear, and same in a primate, as it turns out, and same in this monitos. Monitos are adorable. They look like Dobie the house elf. They have really tiny ears, and they're little. They're little, they're tiny. They're not much bigger than your thumb. Next slide. Is lemurs. These are a type of lemur. They're actually a primate. They learn like humans. These live in France, where they live in a chateau, actually, just south of Paris. And there's a huge facility for them. They grow up in trailers, and they're endangered in Madagascar, but they're overproducing in France. You know, France is the cigarette smoking, the rampant sexuality. So it's one thing to say, Ken, we're never going to be able to transplant human organs because you work on ground squirrels. Try to move along. Stop blocking the road. But these are primates. They can turn off their metabolism completely. Now I can tell you how they regulate metabolism because we know all the tools. We have all the kinase tools. We have all the proteome tools. We have all the DNA sequence tools. We have everything we need now to look at a primate. Overall, if you want a general principle, it's easier to turn off and reorganize a primate than it is a ground squirrel. And here's why. Most of the changes in a ground squirrel are related to changing temperatures so strongly. When you take a mammal from 37 to 5, if I took you down to 5 degrees and infused you with ice cold blood, you just die. Most of the changes in the cold mammals that we've studied forever are because of cold. As soon as you have a warm animal, you can see our papers on bears. As soon as you have a warm hibernating animal, way less changes because you're only dealing with hibernation. So the changes we see in lemurs straight up match humans, straight up match high temperature preservation. And this has changed the field of preservation. There used to be a lot of human organs cool down and then put on ice and then run through, and now they're not. They're put on a machine, they're kept perfused with blood in the warm, they're turned off in the warm, and then you just move them from place to place. So the lemur, and to a lesser extent the bear is a space model. Ooh, bears in space. So the next slide tells you about the lemur, the primate, which is much more like you. They stay warm. They're in a warm temperature. When they hibernate and turn off their metabolism, they stay warm. Hell, it's warm in Madagascar, but it's dry. So instead of going down to 0, instead of being an Arctic ground squirrel, they stay warm. They stay warm like a bear. Remember, bears went down from 37 to about 30. Same with lemurs. They go down from 37 to 30. They're really closely related to us. It's crazy how we're just primates. And so instead of having the complex biochemistry, instead of having to cool something down, I still find cryobiology exciting. These tell us way more about turning off human organs and then reviving them than the ground squirrels did. So the next slide. Uh-oh, we've lost our little stars at the bottom. Oh, how did that happen? This is body temperature and air temperature. So they start out at 37, and even when they're hibernating, they only go down to 32. Look at the black bars. The black bars are their metabolic rate. You have a metabolic rate of, say, 100. You can drop that to a metabolic rate of 98. You just can't turn off your metabolic rate without dying. Look at the black bars. They go down to 0. This is a primate that's turned off all of its pathways and all of its cells and all of its organs. Those black bars that are really near the bottom are essentially 0 metabolism. The only reason why they stay warm is their air is warm. They cool down like a brick to room temperature, whatever room temperature you put them in. It's amazing. It was unknown before lemurs were studied. And the groups we work with in France and in South Africa are really good with animals and organization. And then we run in and do the metabolic underpinnings, which is very exciting. Next slide. If you are interested in the papers, there's always lots of papers. Our students are very productive. These papers are done with massive parallel processing. The students can do genomics, microRNAomics. The students can do Luminex kits where they can measure thousands of different reactions at the same time. And what we can do is measure the stress response, signal transduction, all the stress kinases we talked about, the regulation of genes and proteins by looking at message RNA and microRNA, and then looking at metabolism. You turn down metabolism, what fuels do you use? So in a way, you can get 15 to 20,000 measurements all in an organized fashion from students in the course of a couple of months. It's totally different than when I was a boy, where we had to actually suffer for long periods of time to get 2 numbers. Okay, next slide. There's fewer changes. So when we measure everything in a ground squirrel, we know what changes there are. You can see the papers, there's hundreds of them. But when we measure the lemur, there is much fewer changes. They're all in a very interesting direction. Number 1, mTOR. Translation turns off. The insulin pathway is intimately related to turning off other pathways. The main 12 protein kinases all are on, and AMP kinase is critical among them. It increases, it shifts over their fuels. So mTOR, P38, the same thing in a ground squirrel, only many less changes. Much more happens in a ground squirrel. Hundreds of changes to try and reorganize the cell to be at 5 degrees. And so the critical thing here is microRNA. What's going on with microRNA? So the next slide will help show you that. The reason microRNA is important is that you can add microRNAs to animals. So when we study Drosophila, we feed them microRNA. They pick up microRNA and they change their metabolism. So we study Drosophila as a human health model. Old Drosophila, fat Drosophila, they're amazing, and you can feed them microRNA. When we study tissues and organs, we can take those organs out, pour microRNA on them, and microRNA goes right in. You don't have to do anything crazy. You can just change the amount and type of microRNAs by doing straightforward experiments. Also, you can look at the tissues of an animal and measure microRNA with machines. So as much as it's sort of sad that students can't do PCR and measure all the microRNAs anymore, it is so cool that machines can generate more data than you can shake a stick at. We've got even fourth-year students are analyzing microRNA data where it comes from a machine and then you do a little bit of PCR downstream. So what we did with hibernating primates, just like we did with other things, or what's up with that? We just measured all the microRNAs. There's torpor-responsive microRNAs in hibernating primates. We can also add back microRNAs, which we do with a Department of Defense grant at the University of Chicago and with an organ transplant grant at UCLA, where they work on hearts again. We can add them back to the hearts. So there's a paper coming out on microRNAs in the hearts of transplantable humans and pigs. And the microRNAs that change are all involved in turning off metabolism. They're not involved in organizing for the cold. They're involved in a pure change, which we can mimic in humans. So we can take out a human organ from a donor, you can then microRNA its metabolism down, and then you can move it from place to place. You don't have to cool down an organ and run it through in a short time. The lemur has a number of changes, and some of them are absolutely unique that you didn't see in ground squirrels. So there's species-specific changes in microRNAs, and one of them is here in this picture. You won't know the pathway, you're not supposed to. It's called WNT signaling. It's not WTF signaling. That's totally different. So what this is a new pathway that we didn't even know was involved in organ transplant. We found it because all the microRNA changes. You go, hey, why are these novel microRNAs changing? What do they do? They control a new pathway we didn't even know existed. This pathway regulates calcium. So when you turn off an organ, you cannot afford to let calcium run amok. Calcium that goes into the cytoplasm causes cells to die, and calcium that comes into a cell from the outside definitely causes the whole cell to die. And it also reorganizes the cytoskeletal system. So by knowing the microRNAs and grabbing them up, you can see patterns, and these huge patterns in 1,000 different molecules form into pictures, like abstract pictures of what changes and how it changes. Hanan is our student. You may have met her. She is the lecture TA this year of 2200, and it's been one hell of a year giving a course, like, weirdly from my computer on the 5th floor of Stacy. And then we have the French people that run the lemur colony. They take me out to Paris, and we have pate and discuss lemurs. Okay, the next slide. Is how do you turn off humans? Like, My work is fun and games. We work on weird animals. We turn over a rock. We work on squid that live at the bottom of the ocean, and then you got to go out in a boat and catch a squid. But the thrills that pay the bills are, is this going to be useful? And I think it is. We've identified key processes. You can look at this paper called Bringing Nature Back, Using Hibernation to Change Organ Preservation. Medics are doing this all the time. This is the rage in England and all across the US. We're just part of a huge consortium that just got $36 million. It's called ATP Bio. And we're going to reorganize how organs work. And the critical thing is you get the proper model from nature, and you see it's not that hard at all. So fewer changes. Focus on the microRNA because that's really important. And what follows from the microRNA is all them kinases. What follows from microRNA is histone modification and DNA methylation. So you try to find out what goes on and how you can control it. So next. Yay, there's people. A lot of these people are much more famous than I am. Dustin, Sean, Pierre, Shannon, Kyle, Mike are all professors somewhere. Kyle Bigger's a professor at Carleton. He may have made the wrong choice. We're not sure because Shannon Tessier is now a professor at Harvard. Brian Liu is a medical doctor. Like a lot of our students love to go into med school. I've sent 30 of our students to become medical doctors, and I hope they like me because every time I go to a hospital, there's somebody wanting to stick me with a needle that's taken Biochem 2200. What's up with that? And then we work with people in the US and Canada with the animals. We do not keep lemurs. They keep lemurs in their real facility in France, and then we visit them. The same with ground squirrels. There's no ground squirrels on the 5th floors of Stacy. The ground squirrels are all at the NIH facilities. So we're very lucky. There's a lab site. There's a lot of papers. We've been lucky with having the best students, and yay. That's it. Are there any questions?